Welcome to the Musella Foundation's Brain Tumor Awareness Month webinar series. Tonight's topic is Pediatric Brain Tumors, the Duke Experience, with our special guest speaker, Dr. Daniel Landi, who's a pediatric neuro-oncologist and assistant professor of neurosurgery and pediatrics at Duke. Go ahead.
Right. Thanks, Al, and to the Musella Foundation for including me. It's a pleasure to talk and give this webinar. The purpose of this, from my view, is really just to share what I think are sort of considerations and questions that patients and families have around particularly the clinical trial space of pediatric brain tumors. And so the way that I've sort of outlined my talk, what I'll spend the next probably 30 to 40 minutes on, is to talk about what I perceive are some unmet needs in pediatric brain tumors. I'll talk in a broad sense. First, just what is a clinical trial? What are the considerations around framing a clinical trial? And what either a provider or, more importantly, a patient or their family might consider when pursuing a clinical trial. At Duke, we specialize in particular in immunotherapy, and so I wanted to share some of my background specifically for immunotherapy applications in pediatric brain tumors. I would give an example of a couple notable clinical trials just to go through some of the framework that we've laid, and then talk in particular about some resources and philanthropy of which we're stewards here at Duke, in case that's helpful for anybody watching. As a disclosure, I served on an advisory board for focused ultrasound, which is not something I'm discussing today. Then I would just add, obviously, that there are many thoughts and approaches for how you might treat or engage for a child with a brain tumor. So these are my thoughts, and they're not meant to be definitive or exclusionary to anyone else's. So I think, one of the things that I enjoy in being a pediatric neuro-oncologist, aside from caring for children, is the wide diversity of tumors that we treat. It's a very complicated and intellectually demanding and dynamic field. You can see here that this is just a simple pie chart, which probably needs to be updated. But if you reconcile against a modern pathology lexicon like the WHO, then there are many types of brain tumors, and some we're pretty good at treating, many we're terrible at treating. But there's a wide group, and within each of these subgroups, as we understand more and more about what makes these tumors develop, the molecular and genetic underpinnings are different. So as you talk about how best to treat a patient or a child with a brain tumor, in many cases, we look to the genetic and molecular drivers, but unfortunately, all too often, our actual treatments lag. But there's many different types of brain tumors. If I think about what are the urgent unmet needs as we have clinic, today we run clinic, sort of what are the most difficult tumors that we face or that our patients face? I think obviously high-grade glioma across the board is a large unmet need. The standard approach for these patients is usually surgery, often aggressive, and then radiation followed by some manner of adjuvant therapy. In some cases of high-grade glioma, like diffuse midline gliomas, there's been no effective therapy that's been discovered, perhaps besides radiation. Ependymoma is a glial tumor that arises typically along the lining of the surface of the brain or spinal cord.
Wait a second. I'm sorry, but we're having trouble with the sound. As you move back and forth, when you get close, it's perfect. As you're further away, it's too low.
Okay, thank you.
That's perfect.
Okay, thanks. Yeah, speak up. I'll try to sit really still.
Perfect.
Ependymoma is a tumor that is treated standardly with surgery followed by radiation. It looks as though there's some forms of ependymoma for which adjuvant chemo is helpful, but an ependymoma that recurs after upfront surgery and radiation is very difficult to treat and represents a high unmet need. Recurrent medulloblastoma. If medulloblastoma recurs, particularly after the child's received radiation, this is an incredibly difficult tumor to cure. And medulloblastoma is the most common malignant brain tumor in children. So this is a high unmet need in a tumor for which compelling trials lag in particular. Craniopharyngioma is a histologically benign tumor. It is assigned a WHO grade 1, but this occurs in a very difficult part of the brain that affects the child's pituitary function, their vision, their long-term neurocognitive potential, and the only treatment aside surgery is radiation. There are other trials that are coming, but this still represents an unmet need. ATRT is a very malignant embryonal tumor of young children. And then there are others, even the tumors for which most patients do relatively well. There's not one piece of that pie that's completely cured or effectively treated with benign treatments. So if you talk about what is a medical clinical trial, to start at a very basic level, a medical clinical trial is just a research study that tests an intervention in people. And so this is a formalized, strictly regulated process that looks at something that's experimental or unproven. I think that one of the points of frustration, and rightly so, for patients, families, and providers, is these are high stakes. You have a child with maybe an incurable or very severe form of cancer. And these trials often have eligibility requirements. They can open and close, sometimes very short windows of enrollment. But a clinical trial is a contractual agreement between a sponsor that's regulated closely between the collaborating, providers, regulated by the FDA, and with an IRB in place. And I'll talk a little bit about this. So why do you need clinical trials? Well, this is a framework whereby you can investigate new treatments. For many pediatric brain tumors, existing treatments are not sufficient or they're very toxic. So you need a clinical trial structure and the safeguards in order to keep patients safe and in order to shepherd and promptly evaluate promising treatments. So a robust and well-done clinical trial is necessary so that you can take something that is promising, that has the potential to help people, keep patients safe, properly monitored while they're doing it, in a center that has an established track record of doing proper clinical research, so that those patients can be properly evaluated, the intervention can be properly assessed, and hopefully moved forward if it's found to be compelling and helpful. So within the different groups, whether you're a patient, a family, a doctor, a sponsor, the institution running the trial, then there's different values and goals that you could have for a clinical trial. This is a slide that I borrowed from the FDA's site. And if you talk about how do clinical trials develop, if you're trying to help children with brain tumors, sort of what is the currency? What's the general framework whereby this process can occur? Well, by and large, everything starts in a preclinical space where you have maybe an understanding of how a tumor is activated or proceeds. You have an intervention or some sort of investigation that happens in the laboratory. It undergoes usually some manner of animal testing. And if there's compelling science and something that seems to have good rationale, you can submit an IND application. This allows you, and if granted, gives you permission to study your investigation in the context of a clinical trial in humans. Those trials proceed. They often start as a phase 1 trial and can proceed to a phase 2 or sometimes a phase 3 trial. And these trials are evaluated for safety, feasibility, and efficacy. And if it appears compelling and there's a need, then the sponsor, or whoever's running the clinical trial, can submit an NDA application. The intent of this is to petition the FDA for an approval, whereby the intervention is able to transfer from a clinical trial context with a particular set of investigators more broadly to everybody who's treating that child. So one of the things that I spend time talking with my patients and families about is what are the different phases of a clinical trial. I wanted to talk about this just to make sure that people aren't dissuaded, in some cases unfairly, from earlier phase trials. Most of the clinical trials in pediatric brain tumors in particular are phase 1 or phase 2 trials. I've had some patients or families say that they're not really interested in a phase 1 trial for their son, their daughter, or their loved one. But in fact, the phase of a trial is an indication, in many cases, just of what the primary statistical or outcome measure is of the study. So phase 1 trial is designed to determine, is the treatment safe and what are the side effects? In some cases with the phase 1 trial, there might be a dose escalation where you're trying to determine what is the dose at which the intervention is reasonably well tolerated without side effects. What's the best way to give the treatment? And is the treatment feasible in some cases? You can have, as a phase 1 trial, and certainly every phase 1 trial will collect information on outcomes. It's just not the primary objective or the statistical goal of the study. But many of the compelling phase 2 or 3 trials started as a phase 1 trial, including in pediatric brain tumors. And what gets all of us excited is when you find a patient who responds, particularly a tumor that's otherwise been very hard to treat. Those are examples of a phase 1 trial where you might have a high degree of excitement and a high capacity to help someone, even though the way the trial is designed and formulated is to establish perhaps the optimal dose and also to confirm safety before proceeding to a phase 2 context. So phase 2, in general, the primary objective is to determine, is the treatment effective? In oncology or pediatric neuro-oncology, you want to know, does it work against the tumor? What's the progression-free interval for a patient who's receiving a treatment? Or what's the overall survival of a patient who's receiving this treatment? Statistically, does this intervention seem to help against the tumor? These trials tend to be larger, but they don't have to be. They also continue to collect information on how the treatment affects the body, and these trials will necessarily collect safety and tolerability data as well. As you proceed then, a phase 3 trial typically compares the investigational treatment against a standard treatment. The standard treatment could be something that's FDA approved or something that's widely regarded as an acceptable approach. In pediatric neuro-oncology, rarely do you get to a point where there's a phase 3 trial. Part of this is because the number of patients needed to enroll is very large. The number of patients with a particularly molecularly defined tumor could be very small, or there's not a standardly accepted treatment for which a phase 3 format is well suited. These trials can also take a very long time, and there's always a pressing need, rightly so, to rush promising treatments to children. A phase 3 trial is not requisite for FDA approval. There are many instances, including recently with the Novartis study using 2 oral targeted medicines for pediatric low-grade glioma, where an approval can proceed from a phase 2 trial. A phase 4 trial just refers to ongoing evaluation of treatment that's already been approved. This is a drug that's generally in the marketplace and widely available, but still data and information on the treatment is collected. So I think when I talk with the patient or family, obviously, if they're considering a clinical trial, what they care most about is it effective? But because in many cases, there's not a clearly effective trial or there might be different options to consider, other things and values include, is it toxic? How hard will it be on my son or daughter? What about the overall quality of life? In addition to the side effects of the treatment itself, does the trial or treatment or intervention require relocation, frequent doctor visits, financial toxicity? Obviously, you would love for the patient to benefit. In some cases, the patient or their families will consider also, could others benefit? Even if this doesn't help me or my son or daughter, is this something that could be useful for other children with this diagnosis in the future? I think the goals for a healthcare provider are largely similar. Obviously, all of us want to help our patients. We want a clinical trial that's effective. I think we're all cognizant of how quality of life is impacted, both in terms of the toxicity and morbidity of any intervention. We're keenly aware that there are others who could benefit. How do we shepherd our resources? Which trials do we pursue as a community to evaluate that are most promising and have the greatest need? One of the risks we all face for these relatively rare diseases is diluting out our ability to study any one intervention by having multiple trials. I think we all try and identify the things that are most compelling, support and evaluate these particular interventions as quickly as possible. But that is something that's not easy to do, and not all of us will agree on what is best and most optimal for a given patient with a different disease. I think one of the things I'm cognizant of, especially when counseling a patient or family, is what's the timeline of participation? There's some clinical trials where you can start immediately. You can come in, consent, screen, enroll, and begin treatment that day or that week. Some clinical trials, a CAR T cell, for example, even if swiftly done, will often have a production period of 2 or 3 weeks between the time of enrollment or pheresis or generation of a cellular product. I think what's the timeline, how quickly can you get the treatment to the patient is important. I think all of us as physicians or docs in the academic community feel a sense of responsibility also to our institution, our academic community. These generally align with the patient, but we want to maintain our ability to participate in things like consortium. We want to maintain our collaborations with foundations and academic societies. And I think although we all should focus purely on getting the best patient to the treatment, or the best treatment to the patient, then there are, in some cases, other considerations. In some cases, there might be responsibility to a sponsor. In some instances, there could be conflicts of interest, and these should all be disclosed. But these are things that in some cases might pop up. But by and large, the goal always is to get the best treatment to the child as soon as possible in a way that minimizes the quality of life impact for that patient and family. So I think quickly then, suggestions for families, if that's helpful for anybody that's watching this when considering a clinical trial. I think what you'd really like to key in on, in my view, is how promising is the intervention for a patient-specific tumor. I think I get a lot of emails and inquiries and press releases for something that seems very compelling, but might be in a purely preclinical context. Something that's is very helpful maybe in the laboratory, but the question is, has it helped a similar patient? Is it even clinically available? Has it helped a child who has the same type of brain tumor, the same molecular driver as this brain tumor? And how many similar patients have received the treatment, and what is known about the outcomes? I always try and be very honest and open with patients who are receiving something experimental. Either my experience in terms of the toxicity of the treatment, where known the efficacy of the treatment. I think you want to know ideally that this has helped somebody who's very similar to my child or to the patient. I think you'd want to know, how quickly will I know if it's working, and also how quickly can I get it? There's some clinical trials where the design might be more protracted. I think the responsibility is then on the treating physician to advocate for your patient. I believe in some cases, the output of a clinical trial is too long. And in many of these, especially malignant cancers, you can't wait and shouldn't wait 2, 3, 4, 6 months when it seems pretty clear that this is not the best fit or optimal treatment, and you should pivot to something else. In some cases, you want to consider the order of the possible treatments. For example, there might be one treatment that if you were to receive it, or a part of that treatment would make you ineligible for a treatment later. Avastin or bevacizumab is an example. This is less so in the last few years, but Avastin for a long time was a common exclusion requirement for many clinical trials. And so it might be that you wanted to start with trial A, because then you would still be eligible for trial B and C, but you might not be able to go CAB, for example. I think similarly, the eligibility criteria for a clinical trial will generally require a performance measure. How well is a child doing? How are they feeling? Are they on dexamethasone? And so in many cases, the decision to pursue a clinical trial is best placed early on when those criteria are met, before a child might become sick or with complications from subsequent treatments. I think obviously you also want to consider how hard on the patient will it be. Will it be difficult not only for the patient, but what about the patient's siblings, our family, our overall community? And I think most importantly, what does the patient want? I think in many cases, the children always are very involved, more so I think than we all recognize. Even the very young children are incredibly attuned to not only what's going on with their medical care, but they'll have thoughts and goals too. And I think all of us try very hard to acknowledge and engage the child at their level to understand what he or she wants as best we can. We focus a lot on clinical trials. There are other ways where you can do things that are maybe a bit unproven or a bit individualized that might not fall strictly in the context of an established clinical trial. I think in the contemporary era, certainly at Duke, every tumor undergoes full sequencing. Here we do full transcriptome sequencing on every tumor that comes. And so you can find every tumor, in order for it to develop, it's derived from the rest of your body, but it develops a genetic or an epigenetic change that gives rise to the tumor. And so a targeted agent is just something that's designed to disrupt a particular pathway that's active in the tumor. And so as you have these large throughput whole transcriptome analyses, and these molecularly guided pathology standards that we all use, most recently the 2021 WHO, then you can find and highlight these molecular drivers and see, in many cases, a treatment that might make sense on that particular basis. So when are targeted agents particularly relevant in pediatric neuro-oncology? The perfect example, or what you would prefer or love, is a single pathway, tumor-restricted activating mutation. So you have one pathway that's turned on, and it's turned on only in the tumor. So if you can disrupt that single change in every cell that touches the drug, the only cells that would be affected are the tumor, and that would be sufficient and complete for stopping the tumor. Another way to say that is that the targeted mutation represents an early or initial change that can cause dangerous behavior of the tumor. In some cases, you might have a precursor, an initial genetic change. And if you can catch that tumor at that earlier state, the ability to target that is much more potent and impactful than if that tumor continues to evolve and develop secondary driver mutations. So some of the most compelling and exciting things that have come out of pediatric neuro-oncology in the last several years are some of these targeted therapies. The MAP kinase pathway is ubiquitously and universally activated in pilocytic astrocytoma, which is the most common pediatric brain tumor. It's a benign, histologically benign, grade 1 tumor that causes significant morbidity and sometimes death in our patients. Every pilocytic astrocytoma has a driver in the MAP kinase pathway, many of which are targeted effectively by the MEK inhibitors. Selumetinib and trametinib are non-competitive MEK1/2 inhibitors that are orally administered. There's a liquid version of trametinib that Novartis will start commercially distributing in the next month or so. And these are a good treatment for many of these tumors. There's BRAF inhibitors. A type 1 inhibitor is active against a V600 activated pediatric low-grade glioma. Dabrafenib and also vemurafenib are agents that are effective against these types of tumors. Newer generation pan-RAF inhibitors, such as tovorafenib, which is a type 2 inhibitor, is available as a liquid or a tablet. It's given once weekly, still in the context of clinical trials and expanded access program. These drugs are very successful at targeting these tumors in a way that's convenient for patients and families. The IDH inhibitors, IDH mutations are a driver in proportion of astrocytomas and all oligodendrogliomas. These tumors are a bit more rare in the pediatric age range, but IDH inhibitors, particularly vorasidenib, seem to be very impactful in delaying the progression of these diseases, which are otherwise lethal. Finally, the NTRK inhibitors, entrectinib and larotrectinib, these are activating mutations in a small minority of pediatric high-grade gliomas. They're more common in very young children, but these drugs are very effective when given to tumors that have an activating NTRK fusion, ALK or ROS1 mutation. To get to immunotherapy, again, this is a focus and a strength at Duke. And so I wanted to talk just broadly. I think immunotherapy comes up often, particularly for high-grade glioma, an ependymoma, where there might not be other molecular approaches that are ubiquitous or compelling across many of the tumors. And so in general, immunotherapy is trying to get the immune system to react against the tumor. And so although tumor genetics and epigenetics are relevant, we learn more and more that the genetic and epigenetic environment of the tumor interacts closely with the immune response. But immunotherapy is probably most focused on directing immune cells to recognize markers which are expressed selectively on tumor cells. So, for example, you could target, let's say, an IDH mutant astrocytoma with an IDH inhibitor that is designed to stop the IDH enzyme and the molecular pathway that drives the tumor's behavior. Or you could create a CAR T cell, an oncolytic virus, or a vaccine that's sensitized or targeting to an antigen that's expressed on the surface of the cell, which might not have any role in the tumor activity, but is just a marker that gives the immune system something to hone in on. And so the goal of a cancer immunotherapy then is to generate a T cell response against the tumor. I wanted to spend just a few minutes talking about this because I think as you consider both clinical trials and promising treatments that you could consider specifically for pediatric brain tumors, it's important to know how they're different in a quick sense from other adult cancers for which some versions of immunotherapy are much better suited. This is a quick slide which describes how a T cell works and why the checkpoint, specifically PD-1, is important, but ultimately not generally relevant in pediatrics. So the bottom of this slide shows a T cell, which is a very potent immune cell that's designed generally to fight viruses, intracellular infections, but is also potent against things such as cancers. The T cell natively recognizes an antigen that's displayed on a molecule called MHC. The T cell needs other co-stimulatory molecules to bind and become activated. But natively, because the T cell is very potent, there are self-protective mechanisms in place of important structures in our body designed to shut off a T cell to prevent autoimmunity. You don't want a T cell attacking the gut, the lungs, the thyroid, the pancreas. And this checkpoint is called PD-L1. And so this is a way to shut off a T cell natively that might otherwise cause autoimmunity. However, this checkpoint and others is ubiquitously expressed on many malignant brain tumors and usurped in this way to stop a T cell from attacking a tumor. For some cancers, if you give a monoclonal antibody either to PD-1 or PD-L1, this can be effective at causing the T cell to recognize the tumor and attack it. The problem in particular with brain tumors, specifically pediatric brain tumors, is that the immune infiltrate, the number of T cells in the tumor bed, is very low. And so if you give an intervention designed to cause a T cell to work better and there are no T cells present, then this is not well suited. One of the reasons that this occurs is because pediatric tumors in particular have a very small number of mutations. Usually a very discrete and certain genetic event gives rise to the tumor. But that might not cause the tumor to look very different to the immune system than, for example, an adult tumor, where there are other genetic events that give rise to neoantigens, which are stimulating to the immune system. Melanoma is an example of a very immunologically active tumor, where a checkpoint agent is very effective. But checkpoint agents by themselves that block this PD-1, PD-L1, or other pathway. So if you talk about prevalent or promising immunotherapy approaches specifically for pediatric brain tumors, I put here 3 examples that are a bit different. And I wanted to quickly highlight what the approach is, because there are clinical trials in each of these in the pediatric space, which are helping some patients and which are being studied. So virotherapy, there are a handful of naturally occurring oncolytic viruses, or viruses that naturally bind to and can kill or lyse tumor cells. If you cause inflammation, naturally, what can sometimes happen, if you have inflammation somewhere in your body, that will trigger antigen-presenting cells or other elements to come in and collect the debris, traffic to an immune organ like a lymph node, and talk to a T cell. So the modified poliovirus is an example of an oncolytic virus, binds natively to a molecule called CD155, which is ubiquitously expressed on malignant glioma. It's also expressed on antigen-presenting cells, and when poliovirus binds to these antigen-presenting cells, it can activate it. And so if you give poliovirus, let's say to a glioma cell that expresses the poliovirus receptor, this is natively a cold tumor. There's no immune infiltrate. The poliovirus, as an oncolytic virotherapy example, is very inflammatory. It kills some of the tumor cells, induces neutrophils and other innate immune cells to come in, which then stimulate antigen-presenting cells to come in, collect parts of degraded tumors, and talk to T cells in a way that activates them, shows them the piece of the tumor which has been destroyed, and the hope here is to develop, again, an anti-tumor T cell response that's looking for the tumor. So in this way, you've converted a cold tumor without any T cell or immune infiltrate into a hot tumor. You've done this by causing inflammation as the first step to generate an immune response. Limitations of virotherapy quickly. Obviously, the goal is to stimulate the host immune system against the tumor. Any virus by itself is probably not potent enough to cause sufficient tumor killing in its own right to be clinically impactful. What you need is to damage the tumor, create inflammation, epitope spreading, and prime an endogenous host immune response. So viruses themselves are immunogenic, and they can be cleared quickly. They're something that are generally given directly into a tumor. I'm not talking about the capacity of viruses to serve as vectors, as a form of gene therapy, but you can implant in a virus a particular set of genetic instructions, which can again be piggybacked on to formulate or develop other parts of the immune response. This is a slide I wanted to talk a little bit about vaccines. There's two main types of vaccines that are being used in pediatric brain tumors or cancer more broadly. One is a dendritic cell vaccine approach, the other is a peptide approach. This is a slide that I've taken from a very good Nature Review article that gives, in general, the process whereby a dendritic cell vaccine could be created. So you have a patient who in this case has a cancer. You collect a large number of circulating peripheral blood mononuclear cells, of which a small minority are able to be developed and expanded into dendritic cells. There's different protocols and ways you can do this. The dendritic cell is something that loads a target antigen or a tumor on the surface. They're professionally designed to teach, prime, or activate a T cell that is specifically looking for the marker that's put on the antigen presenting cell surface. You can package these vaccines in multiple doses and give them to a patient either as a single injection, either intramuscularly, subcutaneously, intradermal, and over a repeated period of time. In this way, you're hoping that you can stimulate T cells inside the patient to talk with the dendritic cells that you've modified outside the patient and loaded with the cancer peptide to go out and search the body to attack the tumor. The peptide vaccine approach is a bit simpler. In its most basic form, a peptide is just an amino acid sequence that's mixed with some sort of adjuvant, and it's injected near a lymph node. In order for this vaccine to do anything helpful for a cancer-bearing patient, the adjuvant needs to stimulate an antigen-presenting cell to come out into the tissue, investigate, and engulf the peptide. That peptide is then processed and given traffic back to the lymph node where it could proceed and talk to T cells. One quick slide on a personalized vaccine approach. Again, this is from a Nature Review article. This is a bit hard for me to explain quickly or as succinctly as might be helpful. Personalized cancer vaccines, I think, are a very eloquent and promising way that patients with cancer could be helped. Because every cancer has a unique set of genetic changes and gives rise to proteins that are only expressed in the cancer cells, if you could identify, isolate, and generate a vaccine containing the peptide specifically in a given patient's tumor, this is a promising approach. One of the challenges with this approach is that the way the peptides are processed is different for every person in a way that depends on something called the patient's HLA genotype. So if I had the same type of cancer as 10 other people, and that cancer had the same genetic driver and created the same protein inside the cell that gives rise to the cancer behavior, the way that my immune system would find, recognize, and attack that protein is different. And that's dependent on the HLA genotype and processing, which is hard to predict. So a computer algorithm and a manufacturing process that takes months to generate is both onerous and costs time and money. But if this can be scaled and done quickly, then I think this is a very compelling approach for vaccines moving. Limitations of vaccines are that cancer patients are usually very immunosuppressed. Even if you can create a vaccine that perfectly sensitizes the host immune system to the cancer antigen, in many cases, the host immune system is too beat up, or even if a T cell gets to the tumor bed, it's so immunosuppressive, the T cells are inactive. Most cancer antigens are not the same between patients. This is the concept of HLA dependence, which requires specific algorithms and are very difficult to scale from one patient to the next. A couple of quick slides on T cell therapy. Adoptive T cell therapy is a similar approach to a dendritic cell vaccine, where you take blood cells out of the body, but in this case, you're isolating and expanding circulating T cells. They can be genetically modified and given a synthetic receptor called a CAR, which takes the cytolytic machinery of a T cell receptor, in this case, the zeta chain, and binds it to an antibody that can be manufactured to recognize any tumor antigen. If you're targeting a tumor antigen that is not intracellular, something like EGFR, which is a protein expressed on the surface of many cancer cells, a CAR T cell construct can be the same for many patients, and this is generally how CAR T cells are done. So you take the antibody fragment that you manufacture, you graft it to the cytolytic machinery of the native T cell receptor, and you can create millions of cells that you can give to a patient over and over, either locally, directly into the tumor, or systemically. So CAR T cells are unrestricted in terms of MHC. They're customizable, and they could be used as a third-party product, although this isn't usually done. But if you give someone else's T cells, in many cases, there's not autoimmunity either for the host or the patient. Limitations to CAR T cells are that you target an antigen that's only present on a subset of tumors, or that there's inducible immune resistance, so that even if a T cell reaches the tumor, it's not affected. So I wanted to spend the last couple minutes, in case helpful, just talking about how to find a trial. There's a lot of information out there, and I think if anyone's ever faced or has a child who's faced with a cancer diagnosis, the number of channels and information streams can be overwhelming. I really think, particularly in pediatric neuro-oncology, that the consortia are good places to look. I think three main ones are PNOC, CONNECT, and PBTC, although there are others. In some cases, you might have a compelling investigator-initiated trial at a single institution. We have some of those here. The foundations and patient support groups, including the Musella Foundation, are very helpful in this regard. The Pediatric Brain Tumor Network, the National Brain Tumor Society, and others, in many cases, have truly dedicated, sophisticated, and credentialed people that can help patients and families navigate and get them to a place, a center, a trial, a resource that's helpful. Every clinical trial has to be registered on clinicaltrials.gov, where you can find eligibility, sites, contacts, and a bit about the rationale for the trial. This is usually a good place to look, especially if you're wondering if something's available or if a particular intervention you've read about might be out there. So as a quick example then, if we talk about the PNOC, or the Pacific Pediatric Neuro-Oncology Consortium, these are the sites in the United States, although there are others internationally. And these are institutions that are committed to evaluating promising therapies that are run through PNOC. So we band together. Not every site has every PNOC trial open, but this is a good place to look for, in many cases, compelling, often immunotherapy-based trials for some of the hardest tumors. I think, again, as an example, PNOC has a pretty broad trial portfolio, similar to CONNECT or PBTC, or if you talk about what I consider the high unmet needs are, there's examples of therapies you could consider for each. So for high-grade glioma, or DIPG, there's at least a couple trials. PNOC 022 is something I'll talk a little bit more about. Recurrent medulla, there's a couple trials, craniopharyngioma and low-grade glioma. These aren't nearly the only trials that are out there, but I think as you consider what's something that is compelling, then this is an example of a consortium, a network, a resource that's a good place to start. If we go back and maybe talk about PNOC 022, and I wanted to use this as an example, both as something that I think many patients and families with children who have this disease are considering, but also to go back and revisit some of how we consider, weigh, and evaluate a clinical trial. So PNOC 022 is a trial that's looking at combination therapy for the treatment of diffuse midline gliomas, which includes DIPG. This is a very difficult tumor to treat, and there hasn't been, I'll say, an effective treatment outside of radiation that's been helpful. So this is a phase 2 trial, and it determines if the combination of a new class of medicine called ONC201 with different drugs, currently Paxilosib, is effective for treating patients with these tumors. There's 3 different cohorts depending on when the patient's diagnosed. There's a newly diagnosed cohort, patients who are right after radiation, 4 to 14 weeks, or those who have had progression but before reirradiation, which is something that's commonly done for these tumors. As a phase 2 trial then, the primary objective we've discussed is something like progression-free survival. So for cohorts 1 and 2, the primary outcome measure is progression-free survival at 6 months. The primary outcome measure for cohort 3, those children who have progressed after radiation, is overall survival. So this is an example of a trial that is statistically designed to try and answer this question. But in the case of ONC201, there was a large phase 1 trial. And even though the purpose of that trial might have been initially dose finding, safety, feasibility, the promise and the utility of that drug, even in a phase 1 context, was obviously similar. So this is a trial that's now phase 2, but again, I wouldn't have dissuaded, for example, a patient for whom the phase 1 trial was a fit. It's just different ways that the trials are structured, in many cases, depending on how far along they are. I think one of the things that I wanted to highlight, and something that I think is important that people know about, is something that's a resource at Duke. I think when you talk about clinical trials, in particular the consortium, you're talking about travel, and in some cases it might require travel across state lines or outside of your medical home at your home institution. And so many of our patients have Medicaid as their primary insurer, state-based coverage. And one of the challenges is getting out-of-state Medicaid quickly approved for the ability to travel to another institution to receive medical care, which is especially important with an aggressive or dangerous disease. So at Duke, we're fortunate to be supported by a tremendous philanthropy board. We have a business executive committee, and we're the stewards of a large pool of money whose earmarked purpose is to offset and absolve out-of-state Medicaid. So Duke is able to accept in real time immediate referrals out of state. In many cases, if out-of-state Medicaid is to be approved, it has to be done on the back end. This might take 6, 8, 10, 12 weeks. But we're able to get patients to Duke. We can offset travel expenses, quickly screen them for a clinical trial, if necessary, consent, enroll, and begin treatment. Because this philanthropy, which in some cases can also be used to offset other non-medical expenses, including travel, in some cases, things like bill payment, gas, just all of the many needs that are present for patients when they're struggling with the disease of a brain tumor in a child. And so as I close, I was thinking about how best to sort of prepare this and what the target audience might be. And I imagine everybody who's watching this is committed and tremendously invested in helping a child with a brain tumor. And so I wanted to thank everybody for that and offer anything I could do at any time to help, and in my commitment along with you to help these children. I put this slide, I try and work this slide into all of my talks. This is one of our dear patients. I always get emotional talking about it, but he's passed, and this is when this was Vice President Biden, when the cancer moonshot was coming through. And our patient was sick here, but he made it a point to seek out and was granted an audience with Vice President Biden, including on Air Force Two. And his intent was to make sure that the children with pediatric brain tumors are a priority. And I think certainly the Musella Foundation, many others, and our entire team are committed to rushing, promoting, and helping anything that's promising to our children. So I want to thank all of you for your involvement, and I'll be excited to work forward with you in any way. So I'll close there and we can go to the chat. This is my email. And I think if anybody watching this or who comes across this is interested in a sounding board, you're interested in a clinical trial, you have questions, I'll always try and be very honest with people. I think we try very hard to engage at Duke in a way that's patient-centered, to avoid financial toxicity, and foremost, just to get the best treatment to the child in any way we can. I'll stop there.
Okay, thank you very much. That was very informative. A few questions came in. First one is, what happens with a patient who has no insurance? Could Destination Hope help them?
We can. I think the one, so no insurance is no problem, Al. I think one of the things in huge unmet needs is obviously patients who are international, right? We get many inquiries from patients all over the world. These are children with a serious problem with the brain tumor. And, you know, we don't have the capacity to accept and provide care indefinitely for everybody. We've done that, Al, and I think we try very hard to be judicious in the philanthropy we have. The main purpose of Destination Hope is to offset out-of-state Medicaid during that gap to avoid delays. But I encourage everybody to contact me. The scope of our philanthropy has expanded significantly. And so it's case by case, Al. We do our best, and there's many different ways we can help. So I encourage everybody who's out there for whom finances are a problem to at least let me know. And I know that's a mission of yours too. There are other resources that we're aware of we can work together.
Okay, what are your thoughts on using placebos for pediatric brain tumor trials?
Yeah, I think it's very fraught, Al. I think, like I'd said earlier, most of the even the pivotal studies in pediatrics are phase 2, and so you often don't have a placebo. I think, and rightly so. I think it would have to be a very particular instance where you would consider a placebo. One of the ways you might get around that is with the crossover. There are trials where you can be given placebo. ONC201 had one, the IDH inhibitors had one. But if you are progressive unblinded, then you have the opportunity to at least cross over in some cases and get something that might not otherwise be available. But I'd like to think in many cases that's not something you have to resort to. That depends on the drug, study population, and then ultimately the sponsor and the investigators. So I'm not in favor, but I can understand why it exists.
Yeah, there's the action study with ONC201 is placebo controlled with no crossover.
Yep.
It's not popular.
Yeah, I agree.
Okay, when you were talking about the vaccines and all the immunotherapy stuff, you said brain tumor patients' immune system is not the best to start off with. Does it make sense to use Temodar at the same time as vaccine, which might depress the immune system further? Or would you do the vaccine first and then do Temodar?
Yeah, so Temodar is particularly lymphotoxic. Temozolomide, we usually think in pediatric oncology about big gun chemo and neutropenia and severe immunosuppression. Temozolomide at first glance might not seem that immunosuppressive, but it's really good at killing lymphocytes, including T cells. So you're right, the strategy of giving something that's designed to promote a T cell response alongside something like temozolomide doesn't usually go hand in hand. One of the things we'll often do at Duke, and this is a trick that others do, is if you give something like temozolomide and use it as a lymphodepleter. In other words, let's say you take 5 days of temozolomide and it will suppress the lymphocyte count transiently. There's good evidence that if you give something, a form of immunotherapy, right on the heels of lymphodepletion, that it can be more potent. But in general, if you're doing really immunotherapy, but especially vaccine therapy, you want to avoid things that are lymphotoxic. And that can either just be the tumor itself, Al, or as you progress through standard treatment, the standard treatments are all really bad for lymphocytes. But just natively, the more advanced a cancer gets, the more immunosuppressed a host is or patient, it makes it harder for vaccines or any immunotherapy to work. So I think a general strategy is, can you reasonably move them up front? Can you supplant the standard of care that is inadequate, even if it's temporizing and helpful to some?
Okay, can you say a few words about steroids? There's a couple of questions. Big one is, do steroids help or interfere with the treatment of the brain tumor? Like, I know they help with swelling.
Mhm.
Do they make the treatments work better or worse?
Yeah. So I think in general, steroids are, they can be helpful for quality of life and for function. They're generally not conducive for long-term tumor control, and they're not active against the tumor. I think, examples of where steroids are helpful, let's say that you have a brainstem glioma, and I will consider start steroids specifically if the patient's having a hard time swallowing, or if their balance is so affected that it interferes with their quality of life. And then you can use steroids, usually alongside radiation at the start, to allow them to swallow safely, maintain their balance, and then you wean them off. But steroids, particularly in kids, the main steroid used is dexamethasone. The kids are very sensitive to the side effects of dexamethasone, specifically weight gain, sleep. I've had patients that'll develop striae or thinning of the skin that can weep or ooze. I think, we're a very specialized brain tumor center. All of our patients have brain tumors, and we've received kids who've been on dexamethasone for months and are severely affected from it. So we try very hard, unless the patient is unsafe, at risk of losing vision, or severely affected from a tumor, to stay on steroids for more than a couple, 3 weeks.
What could you use instead to help with the swelling?
Yeah, so I think bevacizumab is a monoclonal antibody against the VEGF receptor. It's something that many pediatric neuro-oncologists don't use it much. We use it often at Duke, and I think many people do in the right context. So we mentioned PNOC 022 as a trial for brainstem glioma or diffuse midline glioma. And in many cases, people getting radiation to the brainstem can have swelling, irritation, in rare cases, necrosis of very important parts of the brain. So Avastin as a steroid-sparing anti-inflammatory IV infusion is allowed on study. So patients on PNOC 022 can get bevacizumab around radiation. And that's a very good tool because, again, you're avoiding the long-term toxicity of steroids, mood, sleep, appetite, anger, with something that is effective at controlling swelling. The risks of Avastin, which include wound healing, it does put you at a slightly increased risk of bleed, blood clots. There's debate about whether it prolongs the course of a disease, particularly astrocytomas. It is FDA approved in malignant glioma in adults after radiation. But in kids, we largely use it as steroid-sparing anti-inflammatory medicine, particularly if you're looking at something like an immunotherapy. We don't want steroids to blunt the immune response.
What about the effects on the growing child itself of Avastin? Does that like stop the growth or anything like that?
Yeah, it's a good question. I think Avastin in the short term, I'll usually counsel patients that, sometimes you can have a hoarse voice. It can make you talk like a hot potato voice. A runny nose is common, but not always. In general, the kids tolerate Avastin really well. The longer you're exposed to Avastin, because it works on blood vessels, it can irritate the small blood vessels in your kidney. So patients who are on Avastin for a long time, this is more common in different types of tumors like low-grade glioma, can develop spilling of protein in the urine and raising blood pressure. The protein in the urine is a hard stop if it reaches a certain point. The high blood pressure usually responds to treatment and isn't a reason to stop. So it can cause problems. It puts you at increased risk of a blood clot. But by and large, it's a medicine that's well tolerated, certainly alongside conventional chemo or some of the other things that we try.
Okay, this one lady asked, she said her child was just diagnosed with a brain tumor. She went to a neurologist, he says it's a brain tumor. He just said, they happen to be in Albany, go to Albany Medical Center. First of all, you don't happen to know any good doctors in Albany, New York, do you?
Yeah, Al, what I would ask is for the patient just to email me and I can, with them, and we can find exactly where things stand, what the needs are.
Okay, then she asked, like, what do you do? Like, you hit with a diagnosis of a brain tumor in a child. I guess the first thing you have to do is get an opinion from a major center.
Yeah.
What else do you have to do?
Yeah, I think the main one, and this is tough. I think the one that you'd love to catch in time is a really good pediatric neurosurgeon. You don't always do that. And, I'm a parent and I can't imagine what that's like. But sometimes by the time a patient or family is diagnosed, they've had a surgery that's either ill-advised or done by somebody without the experience that you would have preferred do it. And so I think if you have a patient who has newly diagnosed brain tumor, especially a child, what you'd love to do would be to get to a very experienced neurosurgeon, a pediatric neurosurgeon. And those, people tend to be at big academic centers with large pediatric brain tumor programs. I think that's relevant for a couple of reasons. Number one, it prevents damage or things that should have been avoided. It also ensures if it's a diagnosis for which you pivot or consider quickly a clinical trial that you maintain eligibility. Some of the PNOC trials require a biopsy be done at an enrolling site with a quick screening consent up front. There are window of opportunity studies where if a patient is safe, you might get something that's investigational for a couple of days before surgery. So I think it just depends how emergent the child needs surgery or what the flexibility is. And if anyone's ever faced with it, to try and get to a really good pediatric surgeon as soon as you can. If not, then look, depending on the diagnosis, it's good, I think, to have a discussion and probably a consultation at a really good academic center specialized in pediatric brain tumors, because you don't know what you don't know. And it's not that any provider, doctor caring for these children is being subversive. They just might not know what the needs are of the patient or what the options are therapeutically.
Okay. Other than the need for a clinical trial, would you say it's worth doing a biopsy for DIPG? Is the risk-benefit ratio there?
Yeah, I do, Al. I think that's something that we recommend for everybody. Again, in the hands of a very good pediatric neurosurgeon. I think this is a procedure that's shown to be safe. And it's important because you can't look at the MRI. You can look at an MRI and diagnose a DIPG, I think, in the right clinical context. But DIPG is not a WHO diagnosis. You could have, I have a 20-year-old with a DIPG that was given that prognosis and information. We biopsied him, and he has a grade 2 IDH mutant astrocytoma. It's still a hard tumor. His prognosis treatment is radically different than a K27 emmet driven. And so I think similarly, when you talk about things like PNOC 022, these are, they intend to be molecularly driven therapies. And so many of the trials will require it. I think that's appropriate. I think the point I make is that the risks are low as long as you have a good neurosurgeon that does it. And then you want to do it in a way where you're optimizing the molecular data. We have protocols at Duke. Our pediatric neurosurgeons take a certain number of cores and a certain orientation. The pathologists know specifically what to do with the tissue. So it's not just that you biopsy it, it's that you then steward and shepherd the tissue to make sure you get the right information out of it.
Okay. Do you know anything about the FLAG 003? It's leg therapeutics. It's going into clinical trials for DIPG soon.
No, not off the top of my head, Al.
Okay, no, that wasn't me. That was somebody else that asked.
Yeah. Have them email me. I can do some research and get back to them.
I think that's just about all the questions we had. We had a lot of questions, but you answered them within the course of that lecture.
Okay.
So
I hope it's helpful. Yeah.
We're going to wrap it up. That's all for tonight's webinar. And we'll see you next time. I'll let you know when the next webinar is. Thank you so much. It was very informative.
Okay. Thanks. Bye.
Bye.